Hello, you're watching Armando Hasurungan Biology and Medicine videos. Please make sure to subscribe, join the forum group for the latest videos, visit Facebook Armando Hasurungan, like, ask questions, answer questions, and please post some interesting things, including some artworks, please. So carrying on from the previous video, we learned how acetyl-CoA was transported from the mitochondria into the cytosol. And fatty acid synthesis, which is the discussion of this video, occurs in the cytosol and begins with acetyl-CoA. And that is why acetyl-CoA was transported out of the mitochondria from the mitochondrial matrix here into the cytosol. So this is really a quick overview before looking at the actual metabolism map. So acetyl-CoA essentially will become malonyl-CoA, either malonyl-CoA or it will become acetyl ACP. Now ACP stands for acyl carrier protein, and this protein helps in the synthesis of fatty acids, which means that malonyl-CoA also will bind to an acyl carrier protein, ACP, becoming malonyl ACP. Now both then, malonyl ACP and acetyl ACP, then join together, join to only one ACP, forming what's called acetoacyl ACP. And this is a mistake. This is not meant to be acetoacetate ACP. It's acyl ACP, also known as beta-ketoacyl ACP, also known as beta-ketobutyryl ACP, and this is done through a condensation reaction. It's got many names, but anyways, beta-ketoacyl ACP then after condensation has three other reactions which we won't look into now, but it's reduction, then dehydration, then it's reduction again before forming acyl ACP. Now this acyl ACP is only short fatty acid ACP, which then leaves the acyl carrier protein and binds to CoA group to form acyl-CoA. And acyl-CoA is still a short fatty acid chain, but by adding carbons and hydrogen with a specific enzyme, it can become a long fatty acid. And also the CoA has to be removed. But back to the map. So fatty acid synthesis begins when acetyl-CoA, this acetyl-CoA which is in the cytosol now, it begins with acetyl-CoA when it can either convert to, so acetyl-CoA can either convert to acetyl ACP or malonyl CoA. Now acetyl ACP is acetyl-CoA without the CoA. The CoA is exchanged with ACP. And ACP, as we have learned, is acyl carrier protein. And acetyl-CoA converts to acetyl ACP by transacetylase. Acetyl-CoA can also convert to malonyl-CoA by a two-step process by this crazy-looking enzyme, and it's biotin carboxylase. Hold up, it's biotin carboxylase and trans carboxylase. So it's a pretty big enzyme, this one. The ACP is added and the CoA is, so the CoA, okay, anyway. Then malonyl CoA converts to malonyl ACP, and with the same enzyme specific for malonyl called malonyl transacetylase, the ACP is added and the CoA is removed to form malonyl ACP. Now, malonyl ACP and acetyl ACP will then combine together to form beta-ketoacyl ACP. And as we've mentioned, there's actually a lot of names for all these things, but I'm just going to keep the acyl ACP in it. So malonyl ACP and acetyl ACP will then combine together to form beta-ketoacyl ACP. And this is by the enzyme beta-ketoacyl synthetase. One ACP was removed, and also carbon dioxide group was removed. And this is a condensation reaction. And interestingly enough, the two carbon addition from malonyl and acetyl ACP to form acyl ACP is a four-step reaction process, as we've discussed: condensation, reduction, dehydration, and reduction. So we have looked at condensation already. The next one is reduction. Beta-ketoacyl ACP is then reduced to form beta-hydroxyacyl ACP by the enzyme beta-ketoacyl reductase. NADPH is oxidized to NADP. beta hydroxyacyl ACP will then be dehydrated, which is the third step, remember, to trans to enol acyl ACP by the enzyme beta hydroxyacyl ACP dehydratase. Dehydratase. Okay. Yeah. Now next, this trans to enol ACP will then be reduced, which is the final step, again, to form acyl ACP. So NADPH is oxidized to NADP because trans to enol ACP was reduced. So that means that NADPH is oxidized to NADP. And this is done by the enzyme enol ACP reductase. So we have just talked about how from malonyl ACP and acetyl ACP, it was condensation, reduction, dehydration, and reduction again. Now, acyl ACP can then switch the ACP with CoA, so the acyl carrier protein is released and the CoA is added to form acyl CoA. Now, this acyl CoA is obviously still very short acyl CoA, which means that it will make a short fatty acid when the CoA is removed. But the short acyl ACP can become a longer chain. It can have a longer fatty acid chain through the addition of carbons and hydrogens through the enzyme fatty acid synthase. So fatty acid synthase adds and makes this acyl group long and then the fatty acid synthase can then also remove the CoA to form the actual fatty acid group without the CoA, because fatty acids are acyl-CoAs without the CoA group attached. I hope that made sense. Please comment, like, and subscribe. So after this, we'll look at how fatty acid degradation works, but first we need to know how fatty acid actually moves into the matrix, because fatty acid degradation occurs within the matrix. Fatty acid synthesis occurs out of the matrix. Thank you.